Yeah, so I thought I would, give an overview today of our, our center, one of the new Phase 2 linked centers at Mount Sinai, and as the name indicates, we are focused on looking at drug toxicity. Okay, so the overall problem that we're looking at with this linked center is drug-induced toxicity. And ultimately, we want to look at 3 different forms of toxicity. Cardiotoxicity, which can take the form of either arrhythmia or cardiomyopathy, which develops over a longer period of time. Hepatotoxicity, which generally leads to a fibrotic liver, or a peripheral neuropathy, which means either pain or numbness in neurons that are caused by drugs. Although what we're focusing on first in the first year of this project is primarily cardiotoxicity. So all the data that I'm going to show are related to that part of the project. Ultimately, we want to do all 3 of these. And what we want to explore with this center is, can cellular signatures of these drug-induced toxicities streamline drug development, and can they predict combination therapies that might have better therapeutic index? And when I mentioned combination therapies, we're faced with this question of which combinations to look at, and we started with the observation, which can be seen, from mining the FDA adverse event reporting system, that multiple drugs, when they're taken in combination, can mitigate the drug-induced toxicity. And this was a study that was published from Ravi Iyengar's lab by one of his graduate students, Sean Zhao, a couple years ago. And what Sean found from mining the FAERS database is that rosiglitazone is a drug that's been pulled from the market, in part because it causes myocardial infarction. But from mining the FAERS database, he found that people who took rosiglitazone in combination with some other drugs, exenatide, metformin, or aspirin, had a lower incidence of myocardial infarction than people who only took rosiglitazone by itself. And you can mine the FAERS database to look at many, many such combinations where 2 drugs, than if 1 drug is taken by itself. And so the overall goal of our link center is to develop cellular signatures for both the initial insult, the toxicity, and the potential mitigation of toxicity when 2 drugs are taken together. The overall experimental computational strategy that we have over the 6 years that we hope to have this center we're going to do experiments on cell types that are matched to toxicity, specifically cardiacmyocytes, hepatocytes, and neurons. We're going to work with both primary cell lines and cells derived from induced pluripotent stem cells that come from well-characterized normal human subjects. We're going to treat the cells with drugs that show adverse events in FAERS and drug combinations that show a reduced incidence of toxicity compared with single drugs. We're going to do several different levels of experimental measurements, gene expression, which we're going to quantify through mRNAseq, protein abundance, which is going to be quantified through mass spec, and then post-translational modification, dynamics and dose response, and we're setting up micro-Western arrays for that. Then I guess what's probably of most interest to this group, since this is a data science group, is multi-tiered computational analysis for signature generation. We want to use both network models, structural models, and dynamical mathematical models, systems of ordinary differential equations, to help us understand toxicity. And then we're going to, once we have enough data and we've performed enough analysis, we want to integrate computational experimental results, and that's going to allow us to test which signatures are the most relevant. Either just lists of genes and proteins, which are statistics, networks, which help provide context, or simulated physiology, which helps us to understand where these systems can break down, where they're fragile. And what I'm going to talk about today, what we've made the most progress on in the first few months of the center, is primarily analysis of the mRNAseq data and integrating this analysis with our dynamical mathematical models. So this is our overall plan, as shown on this slide, but clearly in the first few months, we haven't gotten to all of these points just yet. The overall structure of the signatures in the center can be summarized like this, where we have the experimental layer up here, which, as I just mentioned, is drug treatment, quantification of mRNA, quantification of proteins, experiments on multiple cell lines. We also have some experiments of protein state measurements with the microwestern array. That gives us our experimentally observed signatures. Then down here we have the computational layer where we compute the signatures at different levels of abstraction, network inferred signatures, which are produced through network building, and then dynamical mathematical models and sensitivity analysis gives us these what we call weighted signatures, which are what you get after we perform several layers of computational analysis. The first set of experiments that we're planning to do are experiments with cultured primary human cardiac myocytes, and we're going to treat them with cancer drugs, primarily tyrosine kinase inhibitors, that have known cardiac risk. So this is a list of all the drugs that we're going to test in year 1 here. They all have a NIB suffix on them, indicating that these are tyrosine kinase inhibitors that, as I mentioned, are primarily used for cancer. Through mining fairs, we came up with this list of all these drugs that people may have been taking, for some reason other than the cancer, but it just so happens statistically that when they take 2 drugs in combination, they might have a lower risk of developing cardiomyopathy compared to if they were just taking the cancer drug. So these are the potential mitigating drugs we want to test in combination with these kinase inhibitors over here. And then I'm going to show a couple results that we have towards the end for some experiments with pro-arrhythmic drugs, milrinone, flecainide, and amiodarone, because we want to get some insight into cardiotoxicity due to ventricular arrhythmias, in addition to cardiotoxicity due to cardiomyopathy. So to summarize the progress that we've made so far, we are performing experiments with these cells that we are obtaining from a company called PromoCell. We get lysates from these cells for mRNAseq and proteomics. We have almost completed the experiments with line A, and we've made significant progress on the experiments in line B and line D. With, as I mentioned, one of the main experimental methodologies we're using is mRNAseq. I'm going to discuss this, at some point in the next few slides. But we've done a lot of work on assay development, which has led to significant cost reduction, and this has been done in collaboration with some people at the Broad Institute. And it was actually Peter Sorger, who runs the Link Center at HMS, who was the one who put us in touch with them, the people at the Broad Institute, which has allowed us to do more experiments than we originally thought we were going to be able to do. I'm not going to talk today about the proteomics experiments. That's something else that we're working on, nor am I going to talk about the microwestern array, but this is just to give an overview of all the different things that we're working on in the center. We have also put a lot of effort into generating the standard operating procedures. Some of these are already up on our website. They're going to be up, the rest of them are going to be up on the website soon. And we don't have any mRNA-seq data yet on the experiments with the myocytes derived from stem cells, but we're sort of starting from scratch with the stem cell experiments. So what we've done is we've recruited a bunch of volunteers, run them through testing of their health status, taken some fibroblasts from those cells. We're currently doing the reprogramming so that we can do experiments on the iPSC-derived cardiac myocytes, but that's work that's ongoing. I just want to make sure I acknowledge the people that are doing the work. We've got a team here that are doing a lot of the work in generating the data and doing the experiments. I think special thanks should go to Everin and Tina. They seem to be the ones that are really heading this up. As I mentioned, we're doing these experiments on primary cardiac cell lines from this company called PromoCell. They sell you these cells. You serum starve them, and as you serum starve them, they change their phenotype and they grow into more of a cardiac myocyte-like state, even though I would say they're not exactly cardiac myocytes. So we have to perform some quality control before we're ready to do the experiments. We do a 14 to 28 day differentiation protocol, with differentiation in this case being the serum starving. We want to look at morphology after 14 to 28 days. We can see that when they're undifferentiated, they have an appearance like this. As they spend time in culture, they get bigger, they hypertrophy, and they grow into more of a striated morphology, and they look more cardiac cell-like. We also want to do some immunostaining on them. Before we perform the drug experiments, we do immunofluorescence for alpha sarcomeric actinin. This is a key structural protein in cardiac myocytes that helps form the sarcomeres. In the undifferentiated cells, we can barely see this at all. After a few days, the alpha actinin becomes concentrated at the ends of the cells. That's not exactly what we want to see. This alpha actinin can also be part of focal adhesion complexes. But after 21-day differentiation, this alpha actinin becomes distributed throughout the cells, and that's more of what the phenotype that we're looking for. So this indicates to us that these cells are moving towards more of a cardiac myocyte-like phenotype. And then the other thing that we do for quality control is we do qPCR of known cardiac genes. We have a 7-gene panel of genes that we think should be more highly expressed in the differentiated cells compared to the undifferentiated cells when we initially buy them from the company, including some cytoskeletal proteins, troponin and actinin, a couple of ion channels, and 3 well-known cardiac transcription factors. And we can see that after 14 days or 28 days of differentiation, all of these known cardiac genes go up, and with the exception of connexin 43, which is a little bit of a mild change. All the rest of them go up by at least a factor of 4, indicating that we are getting differentiation towards a cardiac myocyte-like phenotype. Something else we've been working on a lot for the whole LINCS consortium, I would say, and just to make sure that this is well documented and that we can we're going to generate data that people can follow and that's reproducible. We've been writing these standard operating procedures, focusing on metadata, quality control, all these issues that come up in the data working group monthly conference calls. We go through several steps of editing of these protocols before we publish them, and with these protocols we highlight where we do our QA and QC. What are the steps that we have to take? What are the checks that we have to make on these cells and on these data to make sure that they're okay? And then we also highlight where we record metadata on these during this procedure. For instance, an example here is just in growing the cells and plating the cells. We document in these SOPs that we need to take images of these cells. That's our QA QC step, and we need to document that the cells have a particular amount of confluence. And then the yellow here indicates that the images themselves become part of the metadata. So we have put a lot of time into figuring out, what's the right way to document all this. And these SOPs are all up on the website, and we do want to get feedback from people in terms of how, if there's different ways to structure these, what we can do to make these usable, that other people can reproduce the experiments. Okay, moving on now to some of the specific experiments and some of the data that we've actually obtained. One of the things that we spent time developing, or one of the things we've adopted from the literature that I think is going to really help us in these experiments, is performing mRNA-seq with what's known as a unique molecular identifier. This comes from a paper published, I think, in 2012 by a group in Sweden at the Karolinska. And it's a very clever process here. The whole idea is that if you have your mRNA molecules up here, you want to put a unique label on each one of your mRNA molecules. And the reason that you want to do that is because before you perform your sequencing, there's going to be an amplification step, right? You're going to do PCR amplification of your mRNA molecules. But you don't know if a given transcript is going to be amplified once, or if it's going to be amplified 5 times, or if it's going to be amplified 100 times. And that can obviously introduce a bias in terms of your sequencing. And so the whole idea here is that when you do your sequencing, this one here that has like the pink circle on the end of it, you might sequence that particular transcript a dozen times, but because we know that the pink circle only went on once, then you only count this as one unique molecular identifier. And this gray one here, blue-gray one, you also only count as one unique molecular identifier. So that some of these transcripts got amplified a lot, some of them only got amplified a little, but you want to count the unique molecular identifiers rather than counting the total number of reads that you make, and thereby you can get more accurate quantification, and you can get mRNA sequencing quantification that's much less susceptible to biases introduced by the PCR step. And we've adopted this unique molecular identifier, or UMI approach, in a collaboration with a couple of investigators at the Broad Institute. And there's another angle to what they do, which has helped us a lot, which is to have, which we're doing specifically sequencing at the 3' end. And by sequencing in that region only, that's going to be a way to reduce our sequencing costs even further. And with a 96-well plate, they're doing the library prep for $30 a sample. So when we did our first set of experiments, I think that the library prep was costing us a couple $100 per sample. Because they do this in with microfluidics and in a high throughput format, we send them entire 96 wells or 384 wells, and they can do the library prep for much cheaper than what we thought we were going to do, and that allows us to do more experiments, which is good. So we've run a couple of 96-well plates so far. We've really only finished the data processing for the first one. So what we wanted to focus on in our first 96-well plate was a few of the tyrosine kinase inhibitors that might cause cardiotoxicity. We did some of these in combination with several of these mitigating drugs. Of course, we had to test the potentially mitigating drugs by themselves. And then we had several combinations of the offending drugs, plus the potential mitigating drugs. All these on a 96-well plate, and then sequenced all the data. Everything came back all at once. And I should also acknowledge the data analysis and signature team. I'm going to mention a couple of these individuals by name as I go on. But Yugong is a staff scientist, Jens is a postdoctoral fellow who are working a lot with this. Jae-hee and Megan are PhD students, and Joseph Goldfarb is our QA/QC officer. So one of the first questions we had to address when we got our data back is, because we're looking specifically at the 3' end, are you going to be able to map your reads to particular locations in the genome when you're looking at these 3' focused ends? So in other words, how well does the aligner in this case, which is the Burroughs-Wheeler aligner, do in mapping each read? And our reads are about 50 base pairs each. So we did an analysis where what we're plotting on the y-axis is how many genes, and for each one of our genes, we want to quantify what fraction of the reads assigned to that gene are non-unique. So the Burroughs-Wheeler aligner, for every read it has, will tell you, okay, I think this is the most likely location in the genome that this comes from, and I can either say with complete confidence that yes, it definitely belongs to this gene, or I can say, well, it might belong to this one gene, or it might belong to some other gene. And so for every read that we had, and for every gene, we wanted to say, okay, what's the fraction of the unique reads compared to the non-unique reads? And the good news here is you can see that the vast majority of our genes are right down here. The vast majority of genes have 0 non-unique reads associated with it. And then if you look down here at this edge of the histogram, there's a very small percentage of genes where every single read is non-unique. So this particular sequencing protocol is terrible at recording the number, at quantifying expression of these particular genes that have 100% non-unique reads. But with all the rest of the genes in the genome, it's very good at recording that. This is the histogram. This is the cumulative probability function. The idea here being that a small percentage of genes account for 95% of the non-unique reads. So here we're plotting the cumulative fraction of the non-unique reads, and with 5% here, we're up to 95% of our non-unique reads. So the first question we had to address is, if we're only recording at the 3' end, and we're only sequencing about 50 base pairs, is there going to be a problem with uniqueness? And we think there's not a problem with uniqueness because it's a very small percentage of genes that are accounting for all of the non-unique reads that we're seeing. The next question we had to address was, what about reproducibility? Do we have outlier samples? What do we do with these outlier samples? And so we did an analysis with our control samples where we would plot the unique UMI counts for control sample number 1 versus the unique UMI counts for control sample 2. Here you can see control sample 2 versus control sample 4. And the reason to show these two particular examples is here we see a very strong correlation between sample 1 and sample 2. That indicates that this is reproducible. Here we see a bad correlation between sample 2 and sample 4. Indicating that there's probably a problem with one of these. And so what we do is to identify the outliers, we correlate each pair of samples within the same experimental group. Sample 1 with sample 2, sample 1 with sample 3, sample 2 with sample 3, sample 1 with sample 4, etc. And when we compute a Pearson correlation coefficient of every pair in the group, then that makes it easy for us to identify the outliers. So here we have 15 control samples versus 15 control samples, and what we're plotting here in the heat map is the Pearson correlation coefficient. So when each sample correlates perfectly with itself, that's why we have all white along the diagonal here. And what we can see is that the vast majority of our Pearson correlation coefficients are above 0.98, usually above 0.99, with the exception of this red one here, which is sample number 4, which doesn't correlate well with any of the others. And so this is a sort of analysis that clearly identifies sample number 4 as an outlier. And what we'll do is we'll throw this out, call it an outlier, we'll document it, we'll still post the data. Someone might want to dig into the data and figure out what happened, why it's an outlier. But for all the subsequent analysis that we want to do, we'll group all the other 14 samples together after using this criteria to get rid of sample 4. And right now, 0.98 seems pretty good. In future experiments, we want to solidify this QC standard. A complementary way to do this is a technique called multidimensional scaling. This is very closely related to principal components analysis. What this does is compute distances between all pairs of samples and plots the distances between the samples in principal coordinate space. So you get a plot that looks like this, which is your first two principal coordinates, very similar to your first two principal components when you do the analysis that way. And this analysis also identifies this one down here as sample number 4. So this was identified as an outlier through the correlation analysis, also identified as an outlier through the multidimensional scaling analysis. And as I mentioned when I was discussing the previous slide, for subsequent analyses, we group the remaining 4 controls together because they cluster well together, they correlate with one another, with the exception of this one, sample 4. So in the remaining time that I have, I want to discuss what we're doing in terms of analyzing data and generating signatures. And I think one of the things about our center that we're really excited about is computing signatures at different levels of abstraction. And the terminology that we introduced for this is we want to have some signatures, which we're just calling experimentally observed signatures. That's essentially raw data or data with minimal processing. And then other signatures are going to be network inferred signatures. And with the network inferred signatures, we want to leverage some prior knowledge, leverage what we know about interactions between proteins, and generate signatures through those sorts of computations as well. And we can summarize the different signatures that we're going to compute with a table like this, where the simplest thing is a raw experimentally observed signature, and that would be an expression vector for each perturbation. What's the change in gene expression when I add this drug? And then we can get to something a little bit more processed by thresholding and by computing, okay, what are the statistically significantly perturbed genes that I can identify with this perturbation? And so the output of a thresholded experimentally observed signature is going to be sets of, say, the top 50 to top 300 genes. And then from there, we can move to network inferred signatures, which can be enrichment analysis vectors, ranked lists of network nodes, networks of interacting components, which is going to rely on techniques of graph theory, and it is also going to rely on the prior knowledge of how different genes may show correlated expression or how they might physically interact with one another. And then we can move on to processes that require even more intensive computations and also require more prior knowledge, which are going to be obtained through dynamical mathematical models. So the overall way to think of this is, as you go from top to bottom, you go more from the raw data to more processed and simulated data. You also go from totally unbiased signatures to signatures that require more prior knowledge. And I guess the other way to think about this is that the terminology that's come up in some of the data working group meetings is different data levels, right? So up at the top here would be the lowest data level, and at the bottom would be the highest data level. Okay, so I'll finish in the last few slides here with some of the initial signatures that we've obtained through these experiments. One of the first questions we wanted to address after we did our QA/QC is, can we reliably identify differences between different experimental groups? And so we did some principal components analysis of the different drug treatments. What we can see here is that the control samples are all shown in black. Samples treated with sunitinib are shown in green. Samples treated with trastuzumab are shown in blue, and then the ones treated with sorafenib are shown in red. What we conclude from this is that sunitinib doesn't do very much at all. The gene expression after sunitinib seems to be essentially identical to gene expression with control, cells that are just treated with DMSO. Trastuzumab causes some effects, and in some cells, sorafenib in this case causes really large effects. So the sorafenib samples move very far away from the control. And I should give credit to Jae-hee Shim, who's a PhD candidate who's been working with the group and has done a lot of this analysis. Another question we wanted to address is, do mitigating drugs alter the gene expression patterns that are seen with these offending drugs, such as sorafenib? So we picked a set of 150 genes that seem to be differentially expressed in a lot of our groups. This is how we ranked them, from the ones that were upregulated with sorafenib to the ones that were downregulated with sorafenib, and then we compared this pattern of gene expression with what we saw when the cells were treated with sorafenib plus another drug that might mitigate the effects of sorafenib. And we thought this was very encouraging, where the ENT, I can't remember exactly what that drug was, or DIC, didn't really seem to cause much change at all. We see a very similar change in those cases. But URS here, which is ursodeoxycholic acid, did seem to cause a dramatic change in the gene expression patterns when we added this along with the sorafenib. And then DTP caused some change in the gene expression pattern when we added this along with sorafenib. So this does seem to indicate to us that with some, when some drugs are given in combination, it does alter the gene expression pattern. So what we're working on now is calculating statistically significant differentially expressed genes for each group, performing enrichment analysis to identify altered pathways and processes, and then we're constructing relevant networks by connecting the differentially expressed genes through common interacting proteins. I just have a couple more results to show in terms to indicate how far we've gotten with that. We've generated some list of differentially expressed genes. In order to try the numbers the same between groups and to avoid any problems caused by the statistics, we're listing the top 40, top 50, top 100 differentially expressed genes as ranked by significance. These are some that are altered in expression due to sorafenib. These are a few that are altered in expression due to trastuzumab. Probably a more intuitive way to express these results is to do some is to look at processes, look at some pathway enrichment analysis. The Go biological processes that are altered by these top 40 genes in sorafenib are listed here. The ones that are altered with trastuzumab are listed here, and we can see we can see some encouraging things here, such as kinase activity. Right, this is a tyrosine kinase inhibitor here. Protein kinase activity, etc. But probably the most important thing is not these particular processes, but that we're currently working on building the relevant networks. And most important, because of the Links Consortium, is that we're going to release these data by the end of June, with the expectation that other people, when they download the data and start to process it, are going to be able to come up with really creative ways to look at it as well. One other experiment that I mentioned back at the beginning that we're pretty excited about is looking at sex differences in response to pro-arrhythmic drugs. It's well known from clinical experience that women are more susceptible than men to drug-induced arrhythmias and drug-induced prolongation of the QT interval of the electrocardiogram. A lot of these effects are caused by acute block of ion channels. We wanted to see if there was a component of this effect that could be reflected in the altered gene expression. So we treated cells from 3 donors with amiodarone, flecainide, and milrinone. These are well-known pro-arrhythmic drugs, and these are drugs for which there's some indication that they have a bigger effect in women than in men. And the results look like this. This is a principal components analysis looking at the effects of these 3 drugs, but now we're looking at them in 3 different cell lines. So that's why we have 12 unique groups here, control plus 3 drugs, and 3 different cell lines. And what's encouraging here is that these triangles over here correspond to the gene expression that we quantified in a male donor, and these drugs all have mild effects when they're given to the cells that came from a male donor. The asterisks and the circles came from the 2 female donors, and so here with one of these female donors, we see really big effects due to at least 2 of these drugs. So the control samples are the black ones down here, and then the milrinone and the amiodarone are the circles that are up here. Flecainide seems to cause relatively mild effects in this particular female donor, but the other 2 drugs cause much bigger effects in the cells that came from this female donor. So we're encouraged by this. This does seem to indicate that there is some reflection in altered gene expression due to the fact that these pro-arrhythmic drugs cause bigger effects in women than in men. And then the final step we want to take with this involves physiological signatures. What we mean is we want to take these gene expression data and we want to integrate them with dynamical mathematical models in order to predict what the altered physiology is that we're going to see under these sorts of conditions. And the idea here, this is more of like a thought experiment, but it's, I think, probably obvious to everyone that the actual physiological effect that you see depends on which components are perturbed. So looking in a high throughput manner at changes in gene expression is all well and good, but in terms of what effect you're going to see, it all depends on what gets perturbed, right? So you can have one perturbation that causes changes to this group of genes, doesn't cause any changes to this group of genes. And then you could have a different perturbation that cause the opposite effects, right? It might cause strong perturbations to these genes and no perturbation to these genes. And using dynamical mathematical models, that can allow you to predict what the overall physiological effects are going to be. And I picked these 2 particular groups. And even though this is a thought experiment, this is a real simulation. I picked these 2 groups on purpose because perturbation 1 is going to cause a very minimal change to the action potential, whereas perturbation 2 in this case is going to cause a dramatic change to the action potential because the message here is that it depends on what gets perturbed. It depends on which direction they get perturbed, things get perturbed, and how much. But running these sorts of simulations and performing sensitivity analysis is a way to help you quantify the relative importance of the different changes that you see. So we are working on this at the moment. We did this sort of simulation with one of our initial experiments where we added trastuzumab and then we added trastuzumab in combination with ursodeoxycholic acid, and we thought this was a really encouraging result. We'll see if it holds up. But when we looked at the action potential, when we looked at voltage as a function of time in these cardiac myocyte models, we could see that any drug that you gave was going to cause a relatively mild shortening of the action potential, but there was no difference between the potentially cardiotoxic drug trastuzumab or the potentially good drug, the ursodeoxycholic acid, or the combination, right? So the red, the blue, and the green all look the same over here. This is when we're plotting voltage as a function of time. From the same model, from the same simulation, the other main output you might want to look at is calcium as a function of time. So the action potential you care about because that's reflected in the electrocardiogram. Calcium you care about because that's going to indicate how strong the contraction is. And what's cool about this simulation is that control gives a nice strong contraction, a big change in calcium concentration. Trastuzumab causes a decrease in that. And then when you add the ursodeoxycholic acid in combination with the trastuzumab, that's the blue, you see that it goes back up closer to control levels. So this seems to indicate a potential mechanism by which the drug, the trastuzumab, could cause the cardiotoxic effects partly due to a reduction in intracellular calcium. And when the ursodeoxycholic acid is given along with the trastuzumab, that can cause a partial correction of that. So with this first simulation, we see some evidence for mitigation of these effects. Okay, so I just want to conclude then by giving what I wanted to discuss here is just an overview of the center, just to give everybody an idea of what we're working on, what sorts of levels of analysis we want to perform, what the overall goals are, and I should finish by thanking everybody who's part of the team. Mark Birtwistle, Ravi Angar, and myself are the three PIs of the center. Evren, Yibang, Sunita, Jim Gallo, Joe Goldfarb, Millen, Christoph, Avner, Schlesinger. These are all faculty that are involved in the project, and then everyone listed down here are trainees or professional staff scientists who are involved in this. So I thank everybody for being here and paying attention, and I'm happy to take any questions.
So Eric, I have a question regarding this tagging of original transcripts before you run. So presumably, did you guys do any kind of benchmarking in terms of how much noise you remove because you go through this step?
Yeah, maybe I should. I mean, some of that was in the, this is not our method, right? So some of that was in the original paper that described this. But yeah, we have done some benchmarking, as you noted, where especially with some of that correlation analysis, it's what we've done is we said, okay, if you do this on just like the raw read counts, then your correlation between two identical samples or two samples that should be the same is worse than if you do this correlation just on the unique counts. So that's at least one way of quantifying that what we're getting with these unique counts is better than what you get just with more standard sequencing techniques. Yeah, that maybe I should have put that in there, but that's one of the things we've looked at.
I mean, that's interesting. I guess I saw this in your previous presentations, but that's the first time I heard about this technique, and you would think that it would be common, right?
Yeah, I know. I guess so. We are putting a little bit of. Well, one thing. Yeah, I know. I mean, the paper has been cited a lot. The paper that originally developed it, I think it's been cited 80 or 100 times since 2012. But that doesn't mean that everyone who's citing it is using it, right? It could mean that they're just indicating that this is a technique that's out there. We were a little bit encouraged that the Broad is using it, even though the Broad Institute weren't the ones that developed it, even though it was developed in Sweden. So that indicates a certain amount of trust in it. But yeah, it's working pretty well for us. But I agree with you that doing, really documenting what you see is like the increase in accuracy, every step along the way is important.
Hello?
Hello?
Hey, this is John from Stephen's group. I just wondering, could you go to the principal component that plot, which has different colors, which show the principal components? That is previous one? Yeah.
This one or the other?
Yeah. Yes, this one. Thank you very much. Yeah.
Hi. Yes.
Yeah, my question is, this is from this picture. I can see, different technology, different way you match the data. They show, in different clusters. And the question is, can we claim the right one, which is quite unreliable?
Can we claim that which one is unreliable?
The right one is unreliable.
The red one is unreliable. Yeah. Well. At least the red one is causing really big effects. I think what happened with the reason the red one is so much different from the other ones, and right now we're doing the pathway analysis to look at this, is I think the red one, the sorafenib, it just dosed started causing some apoptosis, and started really causing these dramatic changes to the cells. So I don't know that it's necessarily unreliable. I think we're measuring something real that's going on. But I think what happened in these experiments was, all the other drugs besides sorafenib may have been causing minor changes in gene expression, whereas sorafenib was triggering apoptosis or other cell death pathways, and that's why it was causing such different changes.
Mm-hmm.
I think it was, yes, it is causing something qualitatively different, but that does indicate that this is a good method for being able to pick that up.
Thank you. Very, very interesting.
Okay, thank you.
Thanks.
Okay, well, if there are no other questions, Eric, I just wanted to thank you once again for presenting this, and hopefully we'll be interested in analyzing the data once it's released.
Yeah, great. Okay, yeah, and I'm looking forward to the interactions at that level. Once the data are out there, that'll be fun.
Sounds great. Thanks.
Okay, thanks, Mario. Thanks everybody for listening.